I am the Genomics Core Facility Laboratory Manager at Pennington Biomedical Research Center. Probably the first biologist that you've seen today. And this is what biologists are feeling about the data flow right now from next generation sequencing. We're running a solid sequencer from Life Technologies, and it's a short read sequencer. So our questions are, how do we facilitate data analysis to provide meaningful data in a timely manner? And what we need are easily used interfaces between the core laboratory, the principal investigators, and the software provider. We need secure data storage and handling. We would love to have robust and current algorithms that are specific to our experimental applications and which may be quickly and easily updated. We need a choice of alternative algorithms which are designed to analyze application-specific data in different ways. My PIs want to see different algorithms that are either giving them the same story or telling them there's something to look at, that there's something that's not quite right maybe in the analysis. As input, we need algorithms with input file formats, which are the standard for our next generation sequencing instrument, output files, and also file conversion software. And we need output file formats from these algorithms which are suitable for spreadsheet analysis and commonly used internet or server-based tertiary analysis software, such as the UCSC Genome Browser or the Broad Institute software. So I just want to give you an overview of what the flow looks like from raw data to analyzed data. Our raw data is generated by the instrument, of course, and it's generally image files, and that's fluorescent signal in 4 colors. Some of the newer technologies that are coming out may produce numerical signals, though. Then primary analysis is done on instrument, and that's the conversion of the image files to base space or color space. And then we do some basic quality analysis of the data. We take the files off instrument to a cluster or to a compute cloud where we do secondary analysis. At this point, we're going to select our reference sequence. We're going to filter our data. We want to remove primer sequences, ribosomal sequences in our case, and poor quality base calls. Then we want to map our sequences to a reference sequence so we can determine the locations that our sequence maps to. And we want to produce files which are usable for entry to tertiary analysis pipelines. Finally, we do tertiary analysis, and this is experiment application specific. We are currently doing small RNA analysis, chromatin immunoprecipitation sequencing, SAGE sequencing, and exome sequencing. In the near future, we hope to add whole genome sequencing, whole transcriptome sequencing, and methylation sequencing. So we have a broad array of needs as far as our algorithms go. Then we want to see some general quality control information. This is an example of a filter report. We can see the number of reads that we have entered into the analysis pipeline. You can see that that's well into the millions. We can see how many of those reads matched to a specific database and a percentage of that total. Where we have 0 mm, that is a perfect match to the reference sequence. And then we see the number of fragments that match at 1 mismatch, 2 mismatches, or the 3 mismatch level. Additional QC reports that we might like to see would be a read length distribution and also the color composition across the length of our fragment. So now I'm going to give you just a few application-specific examples of tertiary analysis. So I'm going to show you small RNA sequencing, epigenomic sequencing, and exome sequencing. So small RNAs are involved in translation, RNA splicing, gene regulation, genome defense, and chromosome structure. Most classes of small RNA are less than 30 base pairs in length. We can sequence millions of small RNA molecules, and we can quantify the relative frequencies of different sequences in different tissues, developmental stages, or treatments, and determine the statistical significance of the changes. We can track how these expression values change under various experimental conditions. We can map those reads to the genome, and using their characteristics such as length, relative position, or genomic context, we can understand which class of small RNA they belong. We can study the effects of mutations in various genes on the biogenesis of different classes of small RNA. And we can discover new classes of small RNA. So this is an example of some output that you might see from a small RNA pipeline. At Pennington, we are primarily interested in gene expression and changes in gene expression. So this would be a basic chart showing fold change, and that's what the ratio is, and whether that is an up or down regulation in our experiment. We also perform statistics, so we have p-value and adjusted p-value to tell us significance, and we have the gene name, which is a link, and takes us to an outside database called MirrorBase, where we can get detailed information on that particular microRNA. Additionally, we might be interested in families and how similar these microRNAs are to each other. We might want to sort these by expression level, and again, link out to additional information elsewhere. So now I want to give you an example of an experiment that we did that was a small RNA experiment done with next generation sequencing, and also a microarray experiment done with gene expression. So again, we see our table with our information on our small RNA. We are interested in MIR190B. We can click on that and we get additional information about the expression in our treatment and our control group. We took a gene list from our microarray experiment and we submitted that to an online software called GeneSet to microRNA. This software makes a prediction from our gene set which microRNAs may be actively, operating and binding to these genes. We were particularly interested in the coagulation factor 2 gene. And we found when we go to a pathway algorithm that it is an angiogenesis pathway. And so now we've made a biological story that's connecting our small RNA to our gene expression and to a phenotype in our animal. We also do epigenetic sequencing. Epigenetic modification is the control of gene expression through methylation and protein binding, and it's crucial to the understanding of cellular functions. So, for example, chromatin immunoprecipitation sequencing analyzes protein interactions with the DNA. It is used to map global binding sites for any protein of interest. It can be used to determine how transcription factors and other chromatin-associated proteins influence observable characteristics of an organism. Modifications include acetylation, methylation, phosphorylation, and ubiquitination of histones. You can determine how proteins interact with DNA to change the gene expression. I'm sorry, these are a little small. At the top of this illustration, we see the input with the different DNA libraries that have been processed in different manners to obtain this information. On the far left, you see chromatin immunoprecipitation sequencing. The areas of the peaks are the areas where our fragments have detected protein binding. In the center is messenger RNA sequencing. In this case, the peaks are seen at the exomes in the DNA sequence. And on the right, we have methylation sequencing. This is from the UCSC genome browser, And the peaks are showing where methylated and unmethylated sites are in the genome. Thirdly, I'd like to talk about exome sequencing. Researchers are often interested in changes to the genome, such as single nucleotide polymorphisms, insertions, and deletions. Sequencing the entire genome of many individuals is cost prohibitive, but the exome is only about 1% of the human genome. So by isolating the exons from the genome, only the coding regions are sequenced, and the number of individuals who are sampled can be increased in a cost-effective manner. This is an example of a variant list obtained from an experiment that we did recently. It shows the position of the variant on the chromosome and what the base change is at that position. It also shows the number of reads that were stacked in that particular position, a quality score that gives us an idea of whether this change is an artifact or whether it's real, and also a score that shows how many bases the change was involved in. This is an example of exome mapping. So at the bottom we see a reference sequence, and it shows our exons and the location of the untranslated region. Then we see the direction of the read for that particular gene. We see the known SNPs from the DB SNP database. We see the chromosome location. We see the location of the exons, and the color here correlates to which strand those exons are reading on. And we see our mapped fragments, so we can see, since this was isolating the exons, that they are mapping to the areas of the exons. The color here also correlates to which strand these fragments are mapping to. And if we drill down, then we see SNP maps that show us our SNPs and our quality scores, and also give us strand information, so we can see exactly what's going on in that particular location. So this is how biologists are feeling. We're managing, but we're not managing very well. And if I'd known they wanted me to use all of this information, I would never have asked for it. So thank you very much. I'm going to pass the